Now it is my great honor to introduce our second speaker today, Dr. Jacques Côté. Jacques did his PhD at Laval University in Quebec City. From there, he moved to Penn State to join the lab of Jerry Workman for his postdoc, where he conducted some really seminal work characterizing the switch-SNF chromatin remodeler. Following his postdoc in 1997, he returned to Laval to start his own lab, where he hit the ground running with a series of excellent papers characterizing the NUA4 histone acetyltransferase. His lab has then continued to study chromatin modifying complex, how they're targeted to, and how they function on chromatin in the processes of transcription, DNA repair, and replication, and doing this using a combination of biochemistry and molecular biology techniques. Jacques has received numerous awards and accolades. I won't list them all, but I'll just mention he's held a Canadian Research Chair, elected Fellow of the Academy of Science and the Royal Society of Canada, and has been awarded the Louis La Rochelle Excellence Award for his mentorship. All I say, I'm very excited to hear from Jacques today and his talk on MYST acetyltransferases. So with that, please join me in giving a warm, albeit virtual, welcome to our second speaker, Dr. Jacques Côté.
Right. Thank you for a very nice introduction. Everything is fine. You hear me and you see the screen?
Yes, it looks good and we hear you. Great,
great. I'm very happy to be here. I'm very happy to participate in this. I think it's a fabulous initiative that you guys started, and my students and so many students throughout the world are profiting from those interactions, not only from the seminars, but also from the Discord and the community, and encourage you to continue as long as possible to have those activities. I'm very honored to participate. I'm going to talk today about AT complex. You say, yes, we've been knowing about that complex for a long time. And that allows me also to underline the very sad recent passing of David Alice, who identified the first nuclear histone acetyltransferase and made the link to gene regulation. That was absolutely crucial work. He was the giant in the field, and I had the chance to collaborate a bit with him and more with his trainees, and he provided support letters for me during my career. And I think you've seen all on the media, social media, how much people appreciated it, how he was nice, smart, and supportive for so many people. So my lab has been working on histone acetyltransferase complex for a long time. We're not working on GCN5, which was the one identified by Dave, but on a MYST-family acetyltransferase. As you can see here, there's 4 different kind of MYST acetyltransferase and different associated complexes with absolutely different specificity in terms of lysine and histone that get acetylated by those complexes, but they all link to transcription regulation and genome stability. This is a bit too basic for this crowd, but it's just to underline that all the epigenetic marks in the genome are deposited or removed and read by the very often multi-protein complex. And interestingly, those large multi-protein complex often contains multiple reader domain that recognize the different histone marks at different locations in the genome. They also can contain the writers and may sometimes coupled with an eraser in the same complex. So the assembly of those complexes is crucial for the function of those enzyme and reader module. And this is an example of this combination of reader and writers. This is the MOS complex, the CAT6 complex, MOS-morph complex. It's only 4 subunits, but if you look at the reader domain, that complex is pretty amazing. There's PAG domain for K4-trimethyl, PWP domain for K36, a bromo domain for acetone acetylation, a PZP domain to bind nucleosome, a wing helix to bind linker DNA at the exit of nucleosome, and a DPF domain, double PhD domain, that recognize H3 tail as acylated on lysine 14. If you knock out that complex in mouse, you lose completely H3 K23 acetylation in the genome. Interestingly, that there's a crosstalk even between acetylation marks. Like if you mutate HBO1, which is responsible for all of H3K14 acetylation in vivo, as shown by Ann Voss and Tim Thomas' lab in Australia, you don't affect K23 acetyl, but if you look at specific gene, you see that when you lose K14 acetyl, sometimes that specific gene, you lose also K23 acetyl, demonstrating the crosstalk between those two complexes. So Mosmorph recognizing the K14 acetylation at specific loci in the genome and being important for the deposition of K23 acetyl. And that brings me to the point that to study the function of those modifier eraser, you really need to work in the most physiological condition. Yes, you can do things in vivo, but in vivo you always can encounter indirect effect because of the phenotype and stuff like that. So you combine, like we used to do a lot in yeast, combine in vivo and in vitro stuff to understand the exact molecular mechanisms of crosstalk during gene regulation. So in vitro, you need really to work physiological substrate as much as possible because some enzyme just modified freestone, not chromatin. Some enzyme just modified chromatin. They are not able to modify freestones. Some other enzyme, they cannot work on chromatin unless they are native complex. And obviously also with all the reader domain of those complex, you need to have those recognition module also to really recreate what you can see in vivo. And of course, the substrate itself is also important, not only working with peptides or freestone, but also chromatin substrate. And this is just an example that we showed a few years back. HBO1 complex, CAT7, is present in two forms in the cell. In a complex, for tetrameric complex with JAD subunit as scaffold subunit, and with BRPF subunit as scaffold subunit. So this one acetylate H4, and this one acetylate H3. K14 in vivo. If you take those native complex and give them histone, freestone as substrate, they all acetylate H3 and H4 quite equally with no distinction. But if you give them native substrate, native chromatin, purified chromatin, you see that the BRPF complex with HBO1 acetylate H3 tail, while the JAD1 complex with HBO1 acetyl on the H4 tail. Really showing that it is the histone tail specificity is provided by the complex and not by the enzyme itself. And of course, there's other aspects also that have been more recently highlighted in literature, the role of linker DNA in the substrate in chromatin, and also sometimes even the need to have two nucleosomes next to each other to recreate the real function. So with the development of CRISPR, of genome editing, we went on to provide really endogenous complex, not from transgene. We tagged endogenous gene, endogenous protein in K562 cell, and we provide all those MYST families, acetyltransferase complex, tagging different subunit of those complex to really characterize what is the physiological aspect of those complexes, stoichiometry, and so forth. And we had the surprise that when we purified those different subunits of those different MYST family from TIP60, HBO1, Mosmorph, we got all in a subset of purification, the full MLL1, MLL2, KMT2A, KMT2B complex with menin, H2L, and WDR5, but only when we purify the HBO1 JAD complex. So we get it with JAD subunits. We don't get it with PRPF subunit. We don't get it with TIP60 complex, TIP60 complex, and so forth. The full, this was confirmed also by Western blot. So that was a surprise. And this is not on chromatin because this is soluble nuclear extract from those cells. And it's not sensitive to benzonase treatment. So it's not like bridge through DNA or chromatin being co-purifying with these. Of course, most of you know what MLL true thorax complex is. It's an H3K4 methyltransferase. That's very important that counteract polycomb during development. It's also well known to be implicated in 70% of acute myeloid leukemia in child and 10% of all age by a truncation linking it to different partners from the super elongator complex. And it also functions with downstream effector like BRD4 and DUT1A. So if you look at the DepMap website or database, one thing that was striking is that when you look at the codependency over the 1000 and so forth CRISPR screen, you can see that all the HBO1 subunit, JAD, F6, are closely linked in codependency with menin, which is a subunit MLL, and in many cases also with KMT2A itself. Menin itself is codependency with subunit of HBO1 JAD complex. So that was really suggesting that there's a functional link between this HBO1 complex with JAD subunit, the H4 specific one, but not with the H3K14 specific one in vivo. And that brings me to, when you ignore things in your life and you go back and you say, oh, maybe I should have paid attention. So there's a lab in France that contacted us over 20 years ago because they did a trihybrid screen with the portion of the menin protein, which at the time was not very well characterized. It was not known that it was so much functionally linked to MLL. And one of the 5 specific interactors that they discovered was that small portion of the JAD1 protein, the subunit of the HBO1 H4 specific acetyltransferase. And we could reproduce that by just using pull-down assay with recombinant protein. You can see that this small portion of the JAD1 protein is able to interact physically with menin and even pull it down from nuclear extract from HeLa cells. This is just pulling down from nuclear extract, and you can detect that menin is interacting even there with that small portion of the JAD1 protein. We went on to characterize furthermore this interaction in vivo. We used that splice variant of JAD1 that cut that small portion of the domain that putatively interact with menin, and we did lose the interaction with the MLL complex with that splice variant. When you knock down menin, you lose the interaction with between HBO1 and MLL, KMT2A. So that confirms the physical interaction. And then we went on using an HBO1 knockout cells in HeLa cells and looked by ChIP-qPCR the effect of that knockout. And we could see that when you knock out HBO1, of course, you lose the HBO1 signal, but you also decrease greatly the signal of H3K4 trimethylation at different location in the OXA cluster. You lose the menin signal itself. You decrease the MLL signal, and you decrease the downstream effect of DOT1 dependent H3K79 methylation, and you see an increase of polycomb mediated trimethylation in lysine 27. On the other end, when you knock down menin itself, you don't lose HBO1 at the same loci, but you do lose menin itself and MLL. So really arguing that the interaction between the HBO1 JAD complex and MLL through the menin subunit is helping MLL to be located and function at the OX cluster. And just doing RNA-seq, we see a lot of OX gene being downregulated. This is a top hit in the Go term. And when you do cut and run in those cells, the knockout of HBO1, you see that HBO1 is there, the OX-seq cluster, you lose it in the knockout. You look at menin, it's also decreased at specific location, not all location. You see also a decrease of K4 trimethylation. But you also see the striking increase of K27 polycomb mediated methylation, so leading to silencing of those genes. At OXA, it's a little bit more subtle because there's already K27 detected over that cluster, but you see again specific location. So it's not everywhere that you have a dependency of menin and MLL to HBO1, but a very specific location. So to be really true, this physical interaction between MLL and HBO1, MLL fusion protein in leukemia and HBO1 was already identified by the Mark Dawson group in Nature 2 years ago, and also by Aki Yokoyama group in eLife last year. In fact, 2 years ago, we're in 2023 now. What we bring further is that the physical bridge between the two complexes is the menin subunit. And this allows, to favor at specific location the deposition of K4 trimethyl. And downstream effects, since all those ING containing subunits, the MYST-family acetyltransferase that contain ING subunit, have the PHG domain that recognize H3K4 trimethyl. That's why you can see other acetylation marks like H3K14 appearing downstream of the recruitment of MLL and so forth. Now I'm going to switch gear to the TIP16 Neo4 complex. So this is a much larger complex. It has at least 17 subunits, but like those smaller complexes, that multiple histone marks reader domain for a YEADS domain for K27 acetyl, a bromo domain for H4 acetyl, a chromo domain for K36 trimethyl, a PHG domain for K4 trimethyl, an MBT domain for K20 mono and dimethyl. So forks even have an arginine anchor to bind to the acidic patch and so forth. It's responsible for the acetylation of H4, H2A, H2AZ, H2AX in vivo. Most of H2AZ acetylation in vivo is dependent on it. Of course, for H4, there's other that can do H4 in the cell. And it's also has an enzymatic activity that deposits H2AZ in the chromatin. So it has two enzymatic activity, the acetyltransferase for H4 and H2A, and the ATPase, which were one-like activity to deposit H2AZ. And of course, in the genome, often the acetylation and the H2AZ presence correlate. It's important for transcription by many transcript factors like p53, 2F, MYC, NF-kappa B, and so forth. It's also very important for the damage response and repair. It's essential for resection of DNA ends at the double-strand break in combination with the SAGA complex. It's important for autophagy and apoptosis, and it's essential for stem cell, embryonic stem cell maintenance and renewal. We're a lab that initially started working only in yeast. Now we're doing both yeast and mammal. In fact, we're doing more mammals than yeast now, but important aspect that has been reported over the years that the human TIP60 Neo4 complex is in fact a merge of two yeast complex, the Neo4 complex from yeast, which is only has the acetyltransferase activity, is merged with the SWER1 complex, which is the one responsible for deposition of H2AZ. So this is almost an exact merge of those two complexes in the human mammalian TIP60 complex. Of course, there's also the SIRCAP complex that looks like the SWER1 complex in yeast that is responsible for deposition of H2AZ as well. And both purified complex in a swapping assay with H2AZ, where you measure the replacement of H2A with H2AZ on chromatin substrate in vitro, deposit H2AZ in ATP-dependent fashion, both TIP60 and SIRCAP complex. I mean, we did a lot of work on the yeast Neo4 complex, which is a bit simpler than the mammalian one, and it was fun to look over the years the evolution of the structural characterization of the complex, trying to look at how it binds nucleosome, going from the early days of cryo-EM, where in fact this was with Franciscus were mostly the TRA1 subunit that was seen in that cryo-EM, to with Genkai we did the higher resolution cryo-EM at 4.7 angstrom, but it was partial, just a few subunit. With last year report by Eva and Nogales went further with more subunits and higher resolution. And just a few months ago, Zhengshan lab published in Nature even the binding of the yeast Neo4 complex with nucleosome. But again, there's some subunits that are missing, as you can see in the EM in stain model. You see that there's some subunits are missing in the cryo-EM structure still. So there's still work to do. But it was interesting to see the binding to nucleosome, which is still not making so many contacts with the complex. Another aspect that has been characterized over the years in the Neo4 complex, both looking at yeast and human activity, is the fact that the TIP60 or CAT5, the acetyltransferase subunit of the complex, has a chromodomain. So in yeast, we have shown with SONG10 that the chromodomain was important for acetylation of chromatin substrate, not free histones. But of course, everybody thinks chromodomain binds methyl lysine, methylated lysine, K36, K9, K4, whatever. But we've shown, if you look at the structure of the chromodomain in TIP60 and ESA1 in yeast, you see that the hydrophobic cage that's required to bind methylated lysine doesn't exist. It doesn't have the capability to bind a methylated group on the lysine residue based on the structure. And in fact, when you mutate the chromodomain of TIP16 mammals and you test the purified complex, the negative complex with chromatin substrate, you lose specifically the H4 acetylation, not the H2A acetylation and chromatin, but only the H4. And even when you use recombinant nucleosome, which has no histone modification whatsoever, again, you affect the acetylation of the H4 tail. And in fact, that shows that it's a non-canonical role of that chromodomain, and based on the structure that's been published, it looks like it could be interacting with the DNA around the nucleosome, the nucleosome histone interface, and allow the position of the catalytic site of NUR4 near the H4 tail. And just this is a work for Jonathan Humbert. And even we showed that the chromodomain is acetylated in vivo, and then when you mutate to mimic the acetylation of that lysine in the chromodomain, you lose acetylation of the H4 tail specifically, and this could be a very important regulatory mechanism for the activity in a response to DNA damage. So this is clearing a question that, because there's a lot of literature in the field that's saying that TIP60 chromodomain recognize H3K9 trimethyl in heterochromatin, and that's essential for ATM activation in DNA damage response. And we have a lot of data that I'm not showing now that completely contradict this. So the chromodomain doesn't recognize an histone mark and is not required for activation of ATM in vivo. Another aspect that correlate between yeast and human activities is that submodule of NUR4 that also we showed in yeast that exists, the majority of it exists outside of NUR4 and is important for transcriptional elongation to recycle nucleosome histone from in front of the polymerase to behind the polymerase and block spurious transcription. So we call that a small trimer Tintin for trimer independent of NUR4 for transcription interaction with nucleosome. And we recently found from Maya Vadovicu's work in the lab that it is conserved in human. There's a same complex in human that's formed by protein BRD8, MRGPP, and MRG15 that seem to be important for transcriptional elongation. And that also identified the only protein that didn't have a clear homologue in the yeast complex. In fact, E5, in fact, is functionally equivalent to BRD8 in human cells. Now, for the last part of the talk, I'm going to go more in relevant to human disease. So one thing that intrigued us many years ago is the fact that many chromosomal translocation in endometrial stromal sarcoma were in fact fusing or putatively fusing different subunits, up to 7 different subunits of the human TIP16 complex with subunits of polycomb repressive complex, PRC1 or mostly PRC2, in fact. That was very intriguing because that the recurrence of multiple different subunits merged with a polycomb was kind of reminiscent of what happened with MLL, where there's always those translocation MLL with different subunits of another complex super elongator. And so we went on to try to characterize how that worked and what could these fusion create in vivo. And this work was published in Gene Development last year, 6 months ago. So we'll look first at the EPC1 fusion with PHF1. So EPC1 is this subunit in the NUR4 TIP60 complex, and PHF1 is part of the PRC2.1 complex with the polycomb-like paralogs that recognize H3K36 trimethylation to block PRC2 in those locations in the genome. So we purified those fusion protein and the fusion partners from K562 cell, and we found that it is assembling a full mega complex containing both TIP60 NUR4 assembly physically merged to the PRC2.1 complex. It's not partial because when you purify the endogenous subunit of PRC2 or TIP60, we get the other partners. So we get PRC2 coming with TIP60, and we get TIP60 coming with PRC2 when we express the fusion protein in vivo. Of course, this is creating some kind of a monster with all those reader domain and those activities in the cell. We look by ChIP-seq where those fusion would bind. So obviously, the fusion proteins seem to bind to, of course, TIP60 site in the genome, but also to PRC2.1 site and to sites that are bind also by both TIP60 and PRC2. When we look at that H4 acetylation in those cells, we see that we detect an increase of acetylation at the common site of PRC2 and TIP60 and also at PRC2. And this is less clear here visually, but there's also a decrease of methylation of PRC2 target site. One of the most striking places we looked at was, in fact, the OXD cluster, when we could see that when we express the fusion protein versus the partner, we see that when we express the EPC truncated, it binds where you already have acetylation in the OXD cluster. When you produce PHF1, you see that it binds everywhere throughout the OXD cluster, and that correlates what K27 is. When you produce the fusion, you see the appearance of those new acetylation site, and most interestingly, a loss of K27 trimethylation starting from this point. And if you zoom out and look at the IC data, in fact, it's an entire TAD that is affected, not the neighboring one, but this one is affected with overall increase of acetylation and decrease of methylation. This was confirmed by ChIP-qPCR. When you look at that specific location, those specific location of XD13, you see that you have gain H4 acetylation by the fusion. You gain also H2AZ, which is also deposited by TIP60, of course. And you increase transcription of XD13. And this is correlated with K36 trimethylation linked to transcription, and surprisingly, that loss of K27 methylation. It's a surprise because remember, the fusion brings the two activity together. So you still have the acetyltransferase activity, the mega complex, both acetylate and methylate H3K27 in that situation. So in fact, it's explained by the fact that PRC2 is inhibited by K36 trimethylation. So this is shown here with recombinant nucleosome. The mega complex methylate very well, but if you give it nucleosome that are terminated in K36, you inhibit strongly its activity, as it has been reported before. A point that was intriguing is that the majority of those sarcomas, in fact, 50% of the case, implicate another fusion, which is JAZF1 fused with SUS12 subunit of PRC2. So that was intriguing. JAZF1, we purified JAZF1 from those cells by putting a tag expression, and we got exactly the same pattern of band that we usually get when we purified NEO4 TIP60 from human cells. And in fact, this is the mass spec. So in fact, we missed JAZF1 before in all those years because in those cells, K562, JAZF1, for some reason, while being essential in most cell lines, is not expressed in K562 cell. So in fact, JAZF1 seems to be a bona fide stoichiometric stable subunit. Almost all of it is found in TIP60 complex by cut and run and co-localized with all the TIP60 binding site in the genome. And it's interesting because it has been linked to type 2 diabetes, it has been linked to metabolism, to lipogenesis and insulin signaling. So it could be a link to between NEO4 TIP60 metabolism. And in fact, it has an homologue in yeast that's called SFP1, which is a critical regulator of growth that is regulated by the TOR pathway, so nutrient sensing, and regulate the expression of ribosomal protein gene and ribosome biogenesis gene in cooperation with the NEO4 complex in yeast. So that close the loop, so that makes sense. And of course, we're studying more closely all those links in both in yeast and human cells. So the fusion itself, JAZF1 SUS12, we purified it too, and we created the same mega complex specific with PRC2.1, not all PRC2 complex, and again expressing the fusion lead to increased transcription of OXD cluster. So at the end, all those different fusion, 7 different subunits of NEO4 TIP60 are merged in translocation in sarcoma with different subunits of polycomb group complex. And this is the more updated list because it keeps increasing as the year passes by, there's more. And I think that's interesting is that all those are fused with PRC2 or PRC2.1, but this subset of translocation is fused to B core L1 or B core, which is subunit of PRC 1.1, the ubiquitin, the H2A ubiquitin ligase. And those sarcoma seem to be aggressive, high-grade sarcoma instead of low-grade sarcoma in the fusion with PRC2. So obviously, we're studying this to understand why those are high-grade versus low-grade, and that might be linked to PRC1 function different or stronger than PRC2 function in those cells. Importantly, we had access to patient sample, and this is RNA-seq of those patient sample. You see that the tumor tissue versus the normal from the same patient, the normal adjacent tissue. And even though everything we did it so far was in heterologous system, in patient sample, we could confirm that the genes that are activated in those patient sample were mostly genes that were repressed by PRC2 to start with, were containing H3K27 trimethylation, and a big number of OXD and OXA cluster gene that were overexpressed. So confirming the mechanism that we're suspecting, and cut and run analysis of those human samples confirm also the effect. So in normal tissue versus the tumor tissue in the same patient, you can see an increase of acetylation correlated exactly with the decrease of K27 trimethylation in a very polarized or polar way, in the sense that here there's no change, and then suddenly there's a directional change of epigenetic marks. And this is also seen at OXD, so where you see the increase of acetylation, exactly where you see the decrease of methylation. So really confirming that this is the mechanism that's happening. You have the fusion that leads to acetylation at PRC2 target in the genome. So mislocalization of TIP60. It's not mislocalization of PRC2, but mislocalization of P60 that lead to acetylation, leading to transcription, production of K36 methylation that inhibits PRC2 enzymatic activities. And JASF1, in fact, is doing the same thing because it is a subunit of TIP60, in fact. And just as a last point, because the mechanism is a bit related, in acute myeloid leukemia, in another report we published 6 months ago, there's a fusion of Zimin11, which is a protein with critical regulator of active genes that has a PhD bromodomain, PWP domain that recognize specifically H3.3 variant trimethylated on K36. So it's just a fusion of that protein with MBTD1, and that only add that region of the protein to the full TIP60 complex, and that leads to mistargeting of TIP60 to the coding region of active genes, and one of the most important one being the MYC gene itself, and that is purely mistargeting TIP60 by the PWP domain of Zimin11, leading to transcription. If you disrupt the PWP domain, you lose the increase of acetylation, you lose the transcription, and so forth. And this is linked to production of MYC in stem cells, and higher potential of transformation or proliferation of hematopoietic progenitor cell. So, in fact, this mechanism is again mislocalization of TIP60, in this case to the coding region, leading to increased transcription of MYC. And as for sarcoma, that leads to the idea that this may be a way to better treat those specific form of cancer, druggable by inhibition of TIP60. In this case, also blocking PWP domain interaction, or as we published 2 years ago, the interaction with MBTD1 and EPC1, also the interface of MBTD1 with EPC1. So this is, and just finish it. I went a bit over, a lot over, I guess. Sorry about that. So the people who did the work, Sukmin did most of the work on the HBO1 menin physical link and work with the help of Catherine. Dipti and Amel work on the fusion and JASF1 function, and Maeva did the work on the human Tintin and the MBTD1 Zimin11 fusion. And Jonathan worked on the chromosome and TIP60 with the help of Anaita as well, and alumni from the lab, Maria, Karim, Nikita also were involved in the work. And of course, the important collaborator, both locally from mass spec and genome editing and stem cells and bioinformatics. Tanya for the work, a lot of work in collaboration with about the smaller complex. Thank you very much.
Awesome. Thank you, Jacques. That was fantastic. I think we've got, if people want to stay, we've got time for some questions, and there's a bunch that have come in very quickly. I'll remind people you can either type them into the Q&A box or raise your hand and I'll unmute you. To start off with, we've got one from Leanne, who says, "Hi, Jacques, great talk. The requirement of HBO1, JADE1 for MLL1-2 recruitment is very cool. Do you know what is recruiting HBO1 to the Hox cluster? Is there a homologue of JADE1 in flies, and do mutants have a trithorax group like phenotypes?" She says, "I have to run to another meeting, so maybe you can answer these questions over a beer one day." I guess you can look at the YouTube video as well.
That's a very good question. I don't think we have a clear answer how HBO1 would be binding or recruited initially, and that's the main problem with those smaller complexes that have not been linked strongly to a transcription factor dependent recruitment and things like that. So there's a lot of still work to do. I think you have a good question about Drosophila, because I should know the answer for that, because HBO1 in Drosophila is called chameau. Has been linked more to replication and if I remember well, stress response. Yeah, so I don't have a clear answer for you. I'm sorry about that. I think we'll need some beer. I guess. Yeah. I don't know. I don't know. I don't have a mechanism for the initial recruitment of HBO1 JAD on those locations. That's definitely something. I think that there's some data arguing for an F-kappa B linked to HBO1 recruitment, but at the same time, in F-kappa B, TIP60, there's so many, and that's also a problem when you look at all those ChIP-seq of AT complex in the mammalian cell. It's kind of depressing 15 years ago when the first one were published, because all the AT and the HDAC ChIP-seq at all the TSS of active genes, kind of, right? So I think that they tend to be everywhere there's an active TSS, but are they required at all those TSS? I think that's where the key question is. I think there's a lot of redundancy, but in this specific context of Hox cluster, I think we need to continue working on that to figure out what's going on and how it happens. That's for sure.
Jacques, with the fusions, like with EPC1, PHF1, have you looked to see if you get the equivalent of like a piccolo hat module PRC2 fusion, or is it always the mega complex with the full EP100 ChIP60 complex?
Well, that's a very good question, because in fact, if you look at the fusion with the PRC1 complex, there's a truncation in EPC1 that's much shorter in those fusion, and in fact, those shorter version, from what we know, will assemble only piccolo near 4. So we'll not have all the rest, won't have the H2AZ loading module, won't have the other reader module. So arguing that in that sense that it's really the acetylation that's creating the most functional effect in the case of PRC1 fusion. All the PRC2 fusion have the longer version of EPC1, which would assemble the almost full complex.
Okay. So we've got a question here from Sharon, who says, "Really fantastic talk. Do you think the association between the NUA4 TIP60 and PRC2 complexes is linked to the role in maintaining chromatin integrity and stability?"
Maintaining chromatin integrity and stability. You mean in physiological condition, or in the fusion condition? That's what let me read the question again.
I would think under the fusion condition. That's my reading of the question.
Well, it's kind of it's kind of difficult to answer that question, because we have those obviously the most effect we see was always to site where there's already some kind of bivalent chromatin, because there's already some island of acetylation within a sea of K27 trimethylation. So that, because if you look at the TAD next door, which is all PRC2 covered, there's no acetylation there, and the fusion doesn't go there. So obviously the fusion take advantage of already having some island of TIP60 recruitment there, and then the PRC2 moiety allows it to spread over the entire TAD and allowing the change, the functional change. But the question of chromatin integrity, stability, I cannot say. I mean, you're smarter than me, so you might think of more things that I'm able to think at this point.
That's a good answer. And we've got a related question from Nathan asking if the fusions between the polycomb and NUA4 resulted in increase in bivalent chromatin states, or just mostly activating signatures like you see at the HOX-D genes.
Yeah, I would say mostly activating, because everywhere we see an increased acetylation, we see an overall decrease of methylation. So obviously, I'm not saying that it wipes out bivalent, but I'm saying that it's changing the equilibrium. So you still have methylation, but you have more activating marks in the same location. And you know, and I don't want to get in the debate of what is bivalent and if it's and I'm not doing single cell, so that kind of thing. So yeah.
And then there's a question from Thomas who's asking, like he says, fascinating talk, and he was wondering why the transcriptional deregulation you see in the fusion appears to be relatively modest. So kind of like for all the the big changes you see on chromatin, the gene expression changes were not as dramatic.
Well, I think that depends on the because most of the initial work in the first part of the study were done with the cell that were like you know, just producing one copy of the fusion protein in the AVS1 safe harbor. When we use lentiviral expression, then we get a lot stronger effect transcriptionally, and so there's a dosage there, and of course there's also, when you think about the patient tissue and things like that. So in our heterologous system, we have the endogenous white type protein in the background versus the patient sample, which has a translocation, and of course there's a depletion of our normal endogenous protein at the same time. So that might be a reason why some transcription effects are not so big. And of course, depending on the level of expression to start with of those genes at those locations, the full change is going to be changed. It's going to be variable based on the baseline.
That makes sense. Okay, so that's the last of the science questions. Craig Peterson did note after my intro that he would like to claim dual postdoc mentor for you.
Yeah, okay, yeah, sure. In fact, most of the time people think I was in your lab, Craig, because you know, in the switch sniff paper, you're the last one, I'm the first one. So I would say that in the past 25 years, more people were thinking I was in your lab than in Jerry's lab. So you can claim it, and I'll accept it.
Awesome. Okay. So let me just say thank you again to both of our speakers for 2 fantastic seminars. Thank you everyone for attending, and we hope to see you in 2 weeks for the next Fragile Nucleosome seminar.